Good evening everyone. So today we will discuss about antibiotics. I hope you know, and of course you have observed that in last few years question paper almost you will get at least 2 to 3 questions from antibiotics. So here we have to deal with a simple fundamentals, some basic principles of molecular biology and little bit of cell biology, and from here I can assure you that you will get at least 2 to 3 marks. It's your shot, like 2 to 3 questions that your shot you can answer. Okay, so as the name suggests that it's antibiotic, and some of us already familiar with antibiotics, right? Maybe if we face some fever and something, at the time doctor prescribe us to take some antibiotics to kill the bacteria, right? So the major purpose is that to kill the bacteria, right? It can be, you can argue that okay, so in case of viral fever also sometimes it's taken, but the thing is that in case of viral fever there is some other bacterial complication. Just to avoid this antibiotic is taken. Okay, yeah, so just give me, sorry, right, okay, let's minimize it. Okay, now what's the role of antibiotics? First we will start with the antibiotic definition. Antibiotics are chemicals. Okay, we'll proceed with this color. Okay, so antibiotics are the chemical substance produced by microorganism. Remember, it's produced by microorganism, right? Or synthesis derivative that inhibit the bacterial growth or kill the bacteria. So let's say it can work as a both. It can inhibit growth, right? Inhibit growth means it's a bacteriostatic. Okay, right, just give me, sorry, yeah, and it can inhibit growth, or another thing, it can kill the bacteria. That means bacteriostatic and bactericidal. Okay, so we can categorize these antibiotics in different categories based on their mode of action, like how they are actually working. Okay, so first we will discuss about the bacterial cell wall synthesis inhibition. Let's say you know the bacteria has cell wall, right? So what's the difference between gram positive and gram negative? Gram positive and gram negative. What's the difference between the cell wall of these two bacteria? In case of gram positive bacteria, the peptidoglycan is thick, right? Thick peptidoglycan. In case of gram negative bacteria, there is thin peptidoglycan. Thin peptidoglycan, right? In case of gram positive, you can say thick PGN. Another difference that you need to remember in case of gram negative bacteria, outer membrane present, right? But in case of gram positive bacteria, there is no outer membrane. No outer membrane, right? Okay, so we will discuss first about the penicillin, that is quite well known. So target peptidoglycan layer, the main purpose is that the peptidoglycan should not be synthesized. Okay, so if you remember how peptidoglycan is synthesized, let's say we will take one easy example here. Okay, see, there are NAM and NAG, right? N-acetyl glucosamine, beta 1,4, and NAG, N-acetyl muramic acid. Now one pentapeptide is attached here, right? I hope you know, La, L-alanine, the DAP and DA, and followed by the same thing happened in the opposite side, and now these two peptide, okay, they will be linked. This linked is done by one enzyme called transpeptidase. So transpeptidase actually form the this bond, or cross peptidation it can perform, right? So now let's say if you have one drug that can actually inhibit this transpeptidation, then what will happen? Then the peptidoglycan will not be synthesized. So that's the role of the penicillin. So what they do, they actually block the peptide transpeptidase and peptidoglycan synthesis. Okay. Now this penicillin, they have one special ring called beta lactam. Okay, beta lactam antibiotics, right? So how you can see here, these are one polysaccharide chain, this is one polysaccharide chain, and here these two, okay, these two are connected with some maybe pentaglycine chain or maybe some other covalent bond, right? Now this bond, okay, let's say it is penicillin, this is a transpeptidase enzyme, penicillin will come and penicillin has a beta lactam ring. Okay, remember penicillin has beta lactam ring, and because of this, okay, this beta lactam ring will fit here, and now the transpeptidase enzyme is inactive. At this point transpeptidase can't synthesize the peptidoglycan. Now how bacteria acquire resistance? That's also one question, right? So bacteria, they have the beta lactamase enzyme. So I will just zoom out here. Okay, so here you can see actually how bacteria works. Let's say beta lactam antibiotic ring, that's a penicillin, right? Now the penicillin is coming, okay, the penicillin is coming inside the bacterial cell wall. Now let's say this is the penicillin binding protein, okay, inside bacteria. Let's say it's a transpeptidase, okay, and the penicillin can bind with both penicillin binding protein and beta lactamase. If the penicillin bind with penicillin binding protein, then what will happen? Then the bacteria will die. But if penicillin bind with beta lactamase, then what will happen? The beta lactam ring will be clipped. Remember, the beta lactam ring will be clipped, and then the bacteria will not die. That's how bacteria will be resistant. Okay. Now here's some point that you need to remember from here, that the makeup, it's penicillin, cephalosporin, right, or monobactams, these all have the beta lactam ring. That's the most important thing, beta lactam ring. And bacteria, how bacteria got the resistance? Through some beta lactamase enzyme. The beta lactamase actually, they cleave the beta lactam ring, and then bacteria will be resistant. Here the two things you need to remember, right? There's an exam oriented purpose, it's bactericidal. Okay, it's not bacteriostatic. Bacteriostatic means it will slow down bacterial growth, or it will suppress bacterial growth. But bactericidal means what? It will kill the bacteria, and effective against gram-positive bacteria. Mostly effective, right? Why gram-positive? Because gram-positive has a thick outer peptidoglycan layer, right? There is no outer membrane in case of gram-positive. In case of gram-negative, the peptidoglycan, in case of gram-negative, let's say this is outer membrane, this is inner membrane, and one thin peptidoglycan layer is there. In case of gram-negative, inner membrane, this is outer membrane. And for this, somehow gram-negative bacteria got some protection because of this outer membrane. So here what you need to remember in terms of exam point of view, first thing is that bacteria like penicillin, cephalosporin, these have the beta lactam ring. The beta lactam ring, what they do, they inactivate the transpeptidase enzyme. What's the role of transpeptidase? They just connect the peptide, L, La, De, Daf, Da, right? They connect these two peptide, okay, just for to form the peptidoglycan, right? That form the peptidoglycan, okay. Another important point, how bacteria got the resistance, that's to the beta lactamase enzyme that cleave the beta lactam ring of penicillin, okay. Okay, so go next. Okay. Yeah. So inhibition of protein synthesis, that's the most beautiful part. Let's say for any organism, if you can block the protein synthesis part, right, what will happen? If I don't give you protein, what will happen? Your muscle will not be formed, all the things will be stopped working. And the same thing also happened with bacteria, right? So this actually target bacterial Okay, sorry, 1 minute. Bacterial 70S ribosome. I hope you know the bacteria is prokaryote has 70S ribosome, right? And in 70S, what? It divided into 50S and 30S. So what we will do, we will categorize some antibiotic those works on 50S, some antibiotic those works with the 30S. Okay, so first 30S ribosome bind subunit inhibitor, that's aminoglycoside. So they cause misreading of mRNA. Let's say if the ribosome, actually what happened in case of translation, so ribosome is, sorry, mRNA is there, the ribosome come, bind here, they start scanning. Let's say if ribosome read the mRNA wrongly, okay, the reading is wrong, then what will happen? Of course the protein will not be properly synthesized, right? So some aminoglycosides, their name is aminoglycosides, right? What they do, they causes misreading of mRNA. Example is streptomycin, gentamicin. So if you heard this kind of antibiotic name, what they do, they causes misreading of the mRNA. Followed up with this, here you need to, that's also bactericidal and affect aerobic gram-negative bacteria as well, right? Now, if you come to the tetracycline, right, like how tetracycline actually behave, they block the attachment of amino acid tRNA. So if you remember like how mRNA to protein synthesis happened, translation happened. So this is the mRNA, right? So here the codon is there. Then tRNA that has one amino acid in the anticodon, right? Codon anticodon pairing, it will come, right? It will come, and let's say this is 50 and 30. Okay, so what the tetracycline will do, they will block the attachment, okay, at PEA side, right? So first one is P side, then A side, then E side is the exit side, right? So what will happen, the tetracycline, they will mimic this particular tRNA structure and they will block another RNA coming. That's the role of tetracycline. Okay, so that's a broad spectrum antibiotic, and that's bacteriostatic. Remember, tetracycline is a bacteriostatic, not bactericidal. That means it actually halt bacteria's growth. Okay, halt means, let's say if the bacteria is rapidly dividing, okay, in 20 minutes, so it will just tell bacteria, okay, just halt your growth right now. When you will get some favorable situation, at that time you can grow again. Okay, so first one, all these things, let's say streptomycin, gentamicin, tetracycline, they are working on the 30S subunit. Another subunit is the 50S subunit. Okay, how, like there are some macrolides that actually inhibit the translocation. Translocation means, so here if you focus, let's say, yeah, I just want you to focus here. Here you can see there is one side, one tRNA is coming, in the tRNA one amino acid is there and it's coming and occupying here. Translocation means the RNA is here, right? Now the ribosome is coming and scanning. This is called translocation. That means if the ribosome move this direction, what will happen? Now this tRNA will go out, this tRNA will come here, okay, another new tRNA will come and occupy this place. This thing will happen. You understand my point? Translocation means RNA is moving this direction, ribosome is moving this direction. Then what will happen? Of course, one by one tRNA will come and occupy the new space, right? So there are some macrolides that actually inhibit translocation. Example is erythromycin, right? Erythromycin is the greatest example that inhibit the translocation. Chloramphenicol. Chloramphenicol actually inhibit the peptidyl transferase. So if you know how the translation occur, right? So let's say this is one side, this is another side. Now when this tRNA will leave from this particular side, what will happen? It has to transfer this polypeptide chain to this. That's called transferase. So this polypeptide chain is here. Now it need to be transferred to this particular place. That's called transferase, and that's done by peptidyl transferase. Okay, this transfer is done by peptidyl transferase. Now chloramphenicol is the antibiotic that inhibit this transferase enzyme. So remember, there are two categories, one act on 30S, one act on 50S. Okay, if you consider the 30S, there are majorly aminoglycosides, that causes misreading of mRNA. Example is, you already observed, streptomycin and gentamicin. And next is tetracycline, that actually block the attachment of amino acid tRNA in the exact position of let's say E site, P site, A site, something at this A site, right? Example is tetracycline. And the second category is that work on 50S ribosomal subunit, majorly erythromycin, that inhibit the translocation, and the chloramphenicol, that actually block the transpeptidation. Okay, next. Okay, next is the inhibitor of nucleic acid synthesis. Okay, that means, let's say you can block protein synthesis, you can block nucleic acid synthesis as well, you can break the cell wall. That's different mechanism, like how you can kill the bacteria. So bacteria is smart enough to evolve to gain the resistance things, right? First one is the fluoroquinone. Fluoroquinones, it inhibit DNA gyrase or topoisomerase. If you know what is the role of gyrase, let's say during transcription, what gyrase do? Gyrase actually relax the DNA. Sometimes it introduces a negative supercoiling in the sake of replication. It just remove the positive supercoiling things, right? Now, if this positive supercoiling is not removed, at a certain point of time, what will happen? The replication will be halted, right? And if replication is not done, of course cell will die, right? Example is ciprofloxacin. Ciprofloxacin is the gyrase inhibitor. Okay, that is also bactericidal, and it also broad spectrum. If you observe here, this is the ciprofloxacin. Okay, ciprofloxacin is binding with DNA gyrase, topoisomerase, and now these are unable to just conduct the DNA replication. Finally, the bacteria will die. Another beautiful antibiotic is there, that's called rifampicin or rifamycin. Okay, they inhibit the RNA polymerase, majorly beta subunit. Okay, they block this beta subunit. So if you remember, alpha, alpha dash, beta, beta dash, omega and sigma, these are the T7 RNA polymerase, right? So if it bind to the beta subunit, then this core enzyme complex will not form and the transcription will be halted. So fluoroquinone, like ciprofloxacin, it works on replication level, replication, and this rifampicin or rifamycin, they work on transcription level. Okay, it's just shown here. This is binding to let's say there are particular one complex, right? Alpha dash. Okay, let's say alpha dash, beta dash, omega. If beta dash can't bind to the rest of the complex, then T7 RNA polymerase core complex will not be formed. Then it will halt the transcription. I hope it's clear, right? You should be clear. Next one is the inhibitor of metabolic pathway. First one was the cell wall synthesis disruption. Second one is the protein synthesis disruption. Third one was the let's say nucleic acid or like DNA or RNA blocking. Now next one is the metabolic pathway, right? Metabolic pathway means it blocks the folic acid synthesis and majorly sulfonamide. Okay, you see, like from para amino benzoate or PABA, it's known as PABA, para amino benzoic acid. From para amino benzoate to folic acid, from folic acid to folinic acid, and actually the folic acid is the precursor of both purine, and purine, that means a DNA, right? It's from the metabolism. So what the sulfonamide, what they will do, they will inhibit a dry hydrofolate reductase enzyme, dry hydrofolate reductase, DHFR enzyme, they will inhibit, and because of this particular synthesis will not happen, purine synthesis will not happen, folic acid will not be there. Trimethoprim, one inhibitor is there that actually block from folic acid to folinic acid synthesis. Okay, so that means also you can inhibit in metabolic pathways as well, and those are mostly the sulfonamides that block purine or DNA synthesis from the folic acid. Okay, next one and the last one probably. Okay, so this one is the cell membrane disruption. First one was the cell wall synthesis inhibition and this is cell membrane disruption. Those are different things, right? So if you think about the gram-negative bacteria, right? See, effect against gram-negative bacteria. If you think about gram-negative bacteria, they have one outer membrane. Okay, if they have some outer membrane, what will happen? The molecule will come. Okay, right. The molecule will come. Yeah, and once the molecule will come inside, they make some pore. That means once the pore is done, anything can come and come out, right? So osmotic lysis will be done. The main example is a polymyxin B. So the main mode of action is that they disrupt the bacterial membrane. Let's say the membrane is integrity is there, right? Now you just apply something and then membrane integrity is not there, there is some pore. Okay, once the pore is there, anything can come in and come out, right? You can think of some closed vesicle. Once you make some leak, any air, anything can come in and come out, and then it will die. Simple thing, right? That example is the polymyxin B that make the pore in bacterial cell. You can write polymyxin B make pore in bacterial cell membrane. As it works on the cell membrane, that's why the gram-negative bacteria are mostly affected. Okay, so if I want to revise once with you, first one we have the cell wall synthesis inhibition. The best example is penicillin. Penicillin, it blocks the transpeptidase activity. The transpeptidase activity is required for synthesis of the cell wall, right? But the bacteria is clever enough. It is saying, okay, you can kill me, but I also can survive. Through what? It's activating the beta-lactamase enzyme, and the penicillin has beta-lactam ring. So the beta-lactamase will clip the lactam ring, and then the penicillin will be inactive inside the bacteria, right? That's one way. Second thing is the protein synthesis inhibition, right? If you want to inhibit the protein synthesis, either you have to inhibit 70S or you have to inhibit 30S or you inhibit 50S. For 30S, gentamicin, streptomycin, they causes misreading of mRNA. Misreading means, let's say the mRNA will not be properly read. For this, I'm assuming that you know that genetic code, anticodon, right? And I will cover in a few lectures as well. And the next one is tetracycline, that block the attachment of amino acid tRNA. The amino acid tRNA are coming and binding to A site, right? This thing will not happen if tetracycline is there. Next one is the macrolide, that the macrolides that bind to the 50S ribosome, right? It inhibits the translocation. Translocation means mRNA going to this side, mRNA this side, ribosome just scanning this side. Okay, it's just simply sliding movement you can think of. Chloropenicol, it actually inhibits the transpeptidase activity. Okay, transpeptidase means, not transpeptidase, I'm extremely sorry. Peptidal transfer is activity. Peptidal transfer means the peptide is transferring from P side to A side, right? Or the A is coming, P is going to E, so the peptide will be transferred. So this thing will be inhibited. Next one is the nucleic acid synthesis inhibition. First one is the ciprofloxacin, that actually bind to the DNA gyrase or topoisomerase 4, right? So this topoisomerase is actually required for replication, right? It just introduce some negative supercoiling sometimes, or sometimes relax the DNA, because if positive supercoiling is there, then it's very hard for the DNA to be replicated, right? And another thing that transcription level happen, that's a reformation or reformycin, that actually inhibit RNA polymerase. It binds to the beta subunit and block the assembly of the core T7 RNA polymerase and follow up transcription. Next one is the inhibitor of metabolic pathway, right? Metabolic means the basic things. Let's say DNA, purine is not there. Purine need to be synthesized from where? From folic acid. Okay, let's block the folic acid pathway. Let's block the dihydrofolate reductase, right? So that's done by sulfonamide. Next and the last one is the cell membrane disruption. Cell membrane disruption means the mostly affected by the gram-negative bacteria, right? So they have the outer layer, right? They have the outer layer here, outer membrane, and this outer membrane, as outer membrane is there, outer membrane will be easily affected, right? And followed up this some osmotic imbalance, cell will die, right? Great example is polymyxin B. So my humble request to you, please note down all this antibiotic name and their mode of action. Okay. For your convenience, here I have just given everything in a table. Cell wall synthesis. Antibiotic class beta-lactam, best example penicillin. Action binding to penicillin binding protein. Again cell wall, glycopeptide, vancomycin is there. So there are different things. Vancomycin example is different. So they block peptidoglycan. Okay, 30S ribosome. It can be streptomycin, tetracycline, erythromycin. Sorry, erythromycin is 50S ribosome, and again 50S ribosome chloramphenicol. But here it will block the peptidyl transferase. Here macrolide, that means erythromycin, block the translocation. DNA synthesis, ciprofloxacin, inhibit the DNA gyrase. RNA synthesis, basically transcription, are inhibited by rifampicin, inhibit the RNA polymerase. Metabolism purpose, two enzyme, one is sulfonamide, okay, that block the folic acid synthesis that is necessary for dihydrofolate reductase inhibition, followed up with the purine synthesis. And last one is the membrane just disruption. Best example is the polymyxin B. If you remember this table, that's also okay, but please pay attention. You will get at least 2 questions from here. Okay, so that's all about today. Yeah.